Hello, I am Alessandri Luca, PhD student from University of Turin. Here is my project, Sparsely Connected AutoEncoder for single-cell RNA-seq data mining. In order to analyze single-cell data, we developed a workflow called RNAseq, Reproducible Classification and Analysis for Single-Cell Data. This workflow is user-friendly and is provided by a graphical user interface. In the first step, according to the technology used for the FASTQ, you can generate the count metrics that will go through a quality control in the second step, in which you can filter and normalize your data. In the third step, you can pick up a clustering algorithm and classify your data. A very innovative feature implemented is a score, the cell stability score, that will be assigned to each cell and is going to tell you how stable is that cell in that cluster. Removing unstable cell is going to provide a cleaner feature selection, as the feature selection is the follow step, in which you can detect which are the most representative genes for each cluster. The main step of this poster is the last one, in which we use autoencoder and variational autoencoder to do a clustering enrichment and a metagene feature selection. Autoencoder and variational autoencoder are artificial neural network used also for data reduction. We use this structure for a guided data reduction biased with biological information. The neural network latent space is represented by your biological information that can be transcription factor, mRNA, even KEGG pathways. The trick is to use a dense layer that is not fully connected, but only connected to the genes that got a link with the biological information. As validation data, we use the dataset from Zeng, composed by 5 different cell types, FACS purified, that are B cell, naive, natural killer, monocytes, and hematopoietic stem cells. This dataset was in first step analyzed using RNAseq with Griff clustering algorithm, and this provides a clear and stable representation of the data. We trained then the neural network using transcription factor as biological information. The algorithm is going to train several times, obtaining several latent space. These latent space are going to be clusterized in order to estimate 2 important features, the QCM, so the quality control of the method. If the clusters are the same in all the permutation, that means that the method used is stable and the clusters are significant. On the other side, if all the permutation give a different result, so a different cluster, that method is not stable and each cluster is random. Once you have a good QCM, then you can evaluate the second feature, so the QCC, quality control of the cluster. All the clusters obtained in the permutation are going to be compared with the starting cluster, in our case, the one obtained with Griff. If some clusters are stable, that means that that cluster is enriched with the biological information used. Only cluster number 1 and number 2 here got an acceptable value for QCC and QCM. Then we transform the latent space matrix in a frequency matrix, in which we count the frequency of usage of a node, practically how many times that node is different from 0 all along the permutation. Then we fed the feature selection algorithm called COMET with this matrix, and this will provide us the biological information that is most representative for each of our significant clusters. COMET is an enrichment tool that implements the XL minimal hypergeometric test to binarize gene expression data in a gene-specific manner, assessing for each gene and cluster which gene could be a good marker for each cluster. The 4 genes detected by COMET for cluster 1, which is composed mainly by B cell, recapitulate very well some of the key elements of this cell type. The transcription factor PAX5 is essential for the commitment of lymphoid progenitors to B lymphocyte lineage. NF-AT5 is essential for optimal antibody production. THD4 is essential for early B cell development, and RFXANK is involved in activation of MHC2 genes. Moreover, for cluster number 2, which is mainly composed by monocytes, these are the best marker panel made by these 4 genes. This is a COMET output visualization. Dots are the cell, blue color indicate the lack of the 4 genes marker. Red color represents the presence of the 4 marker genes for each cluster. In conclusion, sparsely connected autoencoder can be used as a tool to uncover a hidden feature associated to single-cell RNA-seq data, hidden feature that cannot be seen just looking directly to the genes. The presence of 2 metrics, the QCC and the QCM, provides a measure for the overall SCA model quality. Since ACA analysis would be not so user-friendly for not IT personnel, then the algorithm is implemented in our Class workflow and, of course, is accessible also by graphical user interface. Thank you.